To maintain a stable internal environment, the body must continuously clear out metabolic byproducts. Urine is the vital waste product carrying this load. Normal urine is 96% water, while the remaining 4% consists of a dense solute load, specifically urea, uric acid, and essential electrolytes like sodium and chloride. Its characteristic amber color is derived from a specific physiological pigment called urochrome. To successfully dissolve and excrete the body's daily solute load, the kidneys must produce a strict minimum obligatory volume of 0.5 liters every day. Think of the kidneys like dedicated cleaners tidying up a compound after a busy market day at Akpan Endem in Uyo. They sort through large volumes of debris to discard metabolic garbage while carefully recovering every valuable asset. Survival relies on a non-selective mass filtration of the entire blood volume, followed by the highly selective reclamation of almost every drop of water and nutrient. The nephron is the singular functional and structural unit of the kidney. Each kidney contains approximately 1 million of these units, each responsible for filtering and modifying blood plasma. Blood enters the nephron through the high-pressure afferent arteriole, crashing into the glomerulus, a specialized network of fenestrated capillaries. Large plasma proteins like albumin and red blood cells are too large to pass through the mesh and exit via the efferent arteriole, while the filtered plasma pushes into Bowman's capsule. This chart maps the sequential path of tubular fluid. First, ultrafiltration forces water and small solutes indiscriminately into Bowman's capsule. Next, selective reabsorption in the proximal convoluted tubule aggressively returns essential solutes to the blood. Finally, tubular secretion directly discards metabolic wastes. Filtration, reabsorption, and secretion run concurrently to distill massive volumes of raw blood plasma down to that precise 0.5-liter waste requirement. The proximal convoluted tubule, or PCT, is the primary workhorse of renal reabsorption. As soon as the filtrate enters this segment, the PCT recovers 65% of the filtered water and sodium. More impressively, virtually 100% of filtered glucose and amino acids are captured here via sodium-coupled cotransporters and returned to the blood. Driving this massive volume of material back into circulation requires an immense, continuous supply of cellular energy in the form of ATP. To sustain this effort, the cells of the PCT contain a distinctly high density of mitochondria to power the basolateral sodium-potassium ATPase pumps. Structurally, the apical membrane of these cells sprouts millions of microscopic microvilli, which form a dense brush border. Much like spreading out cassava on a wide mugbata mat to dry under the sun, this brush border maximizes the surface area exposed to transport proteins, increasing reabsorption capacity. Waste products like urea and uric acid are also partially reabsorbed here, as they passively flow down their concentration gradients. The resulting architecture is a masterclass in biological engineering, structurally designed to prevent the lethal loss of essential nutrients. After exiting the PCT, the remaining tubular fluid dives deep into the hypertonic medulla through the loop of Henle. This schematic details the loop's opposing limbs. The descending limb is highly water permeable. As fluid descends, water exits passively, leaving highly concentrated fluid at the turn. The thick ascending limb has inverse properties, impermeable to water, but actively reabsorbs sodium and chloride. It functions like a heavy plastic jerry can. It holds the water inside while actively throwing salt out into the medullary space. Because solutes are removed while the water remains trapped, the internal fluid becomes progressively more dilute as it rises. For this reason, the ascending limb is officially referred to as the diluting segment of the nephron. This contrasting permeability is the mechanism that maintains medullary hypertonicity and dictates final urine concentration. These intense biophysical requirements create a dangerous systemic vulnerability deep within the renal medulla. To prevent blood flow from washing out the delicate osmotic gradient, the vasa recta vessels must maintain an extremely sluggish rate of flow. This flow resembles a heavy traffic jam on Ikot Ekpeni Road in Uyo during rush hour. This sluggish flow delivers very little oxygen to the area, yet the cells of the thick ascending limb have an extremely high metabolic demand for ATP to power their salt pumps. This combination of low oxygen supply and high demand is known as medullary hypoxia. Because these cells operate on the edge of survival, they are highly susceptible to nephrotoxic agents like heavy metals or contrast media, which accumulate in high concentrations in the medulla. The toxic insult causes cellular swelling, necrosis, and the creation of cellular casts that block urine flow. The hyper-efficient architecture required to concentrate urine inherently places the medulla on the brink of physiological collapse, resulting in acute kidney injury. Beyond filtration, the nephron uses tubular secretion to defend the body's narrow systemic arterial pH range of 7.35 to 7.45. During metabolic acidosis, the kidneys act as the ultimate buffering defense. Tubular cells hydrate carbon dioxide. Bicarbonate is reabsorbed into blood as a buffer, while hydrogen ions are actively secreted into the tubular fluid, neutralized by phosphate and ammonia, and excreted. This continuous extrusion of hydrogen ions directly returns systemic blood pH toward normal. Ultimately, the nephron functions as an energy-intensive, highly targeted physiological engine that relentlessly engineers the entire body's chemical survival.